Okay, all right. So I think we can get started. So welcome to our P30 NIH Center seminar series. So today we are pleased to have Dr. Alex Perkins from the University of Notre Dame to talk about mathematical modeling of vector-borne diseases. So Dr. Alex Perkins is an associate professor in the Department of Biological Sciences at the University of Notre Dame. He and his groups applies mathematical, computational, and statistical approaches to answer basic and applied research questions about the ecology and epidemiology of infectious diseases. They worked extensively on mosquito-borne diseases and other vector-borne diseases of humans and animals, emerging diseases and respiratory diseases. So he published extensively on infectious disease mapping, forecasting, intervention, and prevention, and is a leading expert in this area. So today, Dr. Perkins will talk about addressing uncertainty in vector-borne disease models. So without further ado, I will hand it over to Dr. Perkins.
All right, thanks so much for the kind introduction, Sen, and also for the invitation to speak here today. So like Sen said, I'm going to be talking about uncertainty in models of vector-borne disease epidemiology, and in particular, uncertainty around assumptions that we make. So again, all this is about assumptions, which are really central to all models. Models are really just sort of an elaborate concoction of assumptions about how we think the world works. And of course, that can be very powerful for making various predictions and gaining insight into how the natural world works, to have that sort of simplified understanding. But even so, we can go wrong with assumptions. There's this famous saying that when you assume, you make an ass out of you and me. And so that's something that we need to be aware of, and that really sort of underlies my talk, is concern over where we can go wrong with our assumptions. So just to give you a few examples of some models in infectious disease epidemiology that have had assumptions with consequences that are not really desirable, and you could consider these assumptions to be flawed. One would be in some of the work that's been done on pertussis and trying to understand the implications of vaccines and the role of vaccine failure in explaining its dynamics. So some nice work by Jenny Levine and others has showed that if you make arguably the wrong assumption about whether immunity is waning or boosted and all these sorts of dynamics, that can have big implications for what we think about how vaccines work and policy decisions about how they should be used. So this is an area where there's been really stark contrast show up in terms of assumptions. Another is in work on HIV, where there was some controversial work a little over a decade ago that proposed that ART could eliminate HIV within South Africa within 7 years. This had really important policy implications, but other modelers pointed out that there were some really concerning and simplifying assumptions that underlied these predictions, and that when more realistic assumptions were put in place, that the conclusions and implications changed drastically. So yet again, a sensitivity to assumptions that turned out to be important. And then finally, if we think back a little more than 2 years ago, when we were all wondering where things were headed with the pandemic, there were some models out there, this one by IHME most famously, where there were certain assumptions made about the functional form of how an epidemic and its trajectory work that drove a lot of their predictions. And these were taken very seriously when in fact they probably shouldn't have been. Here you see how the uncertainty shrinks, this conveys a sense of very high confidence that the epidemic is going to vanish within a relatively short time frame, when in fact that's really just a consequence of the structural assumptions of their models. So I would argue that this is another really important example of a place where you can go wrong with assumptions. So in dealing with uncertainty in modeling more generally, there are several ways that we tend to think about this and that we can address it. So one that's, I would say, most common is that we think about parameter uncertainty, and we fit or calibrate models to data in order to reduce that uncertainty. So if we think about things like Bayesians and we think about parameters in the model as being uncertain to begin with, and we express a prior distribution to reflect that uncertainty, we want to bring in data to try to narrow down that uncertainty and refine our understanding. So that's one way that we commonly deal with this issue. But this just relies on a single model and confronting it with data and trying to reduce the uncertainty around that model's parameters. If we want to think about how we use multiple models, again, there are a variety of different ways to do this. So I would say a conventional, traditional way to do this in science in general is to use models, whether mathematical or conceptual, to generate competing hypotheses, that make competing predictions, and to go out and do experiments or collect data that somehow inform back on whether those predictions were turned out to be right or not, and then sort of narrow down the model space that way and reduce our uncertainty about these competing models. So again, this is a very traditional approach, and it certainly has its place. If we want to rely more explicitly on models and use models in how we learn about the world, we can do something similar where each hypothesis or set of assumptions maps onto a different model, and then we see how well that model explains a given pattern in data. Again, this is something that I would say over the last decade or two has happened increasingly as the ability to fit models to data has gotten more sophisticated. And then finally, if we want to go even a step beyond that last approach, and rather than just seeing how well alternative models explain data, seeing how well alternative models can make predictions in contexts that the models have not yet seen or sort of been trained to. And so this is really the most challenging approach to test a model, and a potentially very rigorous way to learn about which models perform best. And it at least tells us about performance in a predictive context, which can be useful for many applications of models. So I'm going to make a proposition to you today and show you two examples of work where we have wrangled with these issues. So my proposition is that alternative assumptions and uncertainty in them should be accounted in modeling more routinely. There are plenty of good examples where this is done, but again, I think it's the routinely part that I want to stress, that I think we often think about a single model. We condition our analysis on that. We fit that model and we deal with parameter uncertainty, but we're often not fully exploring this assumption uncertainty. And these assumptions that we should consider and test and challenge range from assumptions about the underlying biology of the system to the mathematical structural form of the model to what the assumptions and interpretations are about data in our models. So there's a whole range of assumptions, any of which can cause problems, as I showed you earlier. So like I said, this work is going to focus, or this presentation is going to focus on two pieces of work we've done. One was in a more spatial context, and one is in a more temporal forecasting context. And I will say before I get into this that, I think these are two examples where I can show you how we did address these issues, but there are plenty of papers I've written where we probably have not fully explored assumption uncertainty. So I'm as guilty about this as anyone. But nonetheless, these were two projects where we were really sort of forced to deal with these issues of assumption uncertainty, even if that wasn't our primary goal. So I hope that you'll learn something from these examples. So the first one, again, this is in more of a spatial mapping context, and it's related to yellow fever. And ultimately, our goal in this work is to predict the impacts of vaccination on the distribution and burden of this disease. So before I proceed further, I just want to thank several folks from my lab who made really important contributions to this work. Some of whom have moved on and others are still here. Quantron in particular is sticking around and continuing to build on the work that I'm showing you today. So look out for more exciting things on yellow fever to come from him in the future. Okay, so like I said, the objective here, first and foremost, is to infer the burden of yellow fever. We're focusing on Africa, which is really the hub of yellow fever's distribution globally. And then once we understand that burden, we want to project the impact of vaccination on reducing that burden of disease. So just a couple of things to note about yellow fever. This is a primarily zoonotic disease, although it does have the potential for transmission in urban settings via Aedes mosquitoes. There's a long history of yellow fever. It is vaccine preventable, given that we're talking about the impacts of vaccination. Other things to note that are very important are that there's a large asymptomatic reservoir. A lot of cases are mild, but there's an appreciable fraction that are severe. And among those severe cases, approximately half can result in death. So it's a quite serious disease. And given these numbers here, you really don't want to have a whole lot of people getting it. So the premise of how we're tackling this issue of inferring its burden is to first use serological data to provide information about underreporting. So we're assuming that serology can tell us about all the infections that are out there whether they're apparent or not. And then that sort of gives us a basis for comparing that to reported cases and then seeing what that multiplier is and adjusting in that sort of way. The other thing that we leverage here are environmental variables that provide information about factors that could be associated with risk of zoonotic spillover related to various environmental conditions that I'll mention later, given that that's really probably the primary driver of exposure and infection as of now. Okay, so I'm going to walk through a series of steps about how our process for estimating the burden and projecting vaccine impact works. So like I said, serology is what provides information about past exposure to the virus and potentially more infections than are reported in surveillance systems. So in general, the more individuals there are with positive serology, the higher we're going to infer the force of infection to be. But one tricky thing is that you can have positive serology either because people have been infected or because they've been vaccinated. And the way that we attempt to correct for this is by taking very detailed estimates of vaccination coverage by age, location, and year, and basically assuming that any seropositivity in excess of that vaccination coverage would be attributable to natural infection. So that's how we learn about the natural infection side of things. And so this just kind of gives you a sense of what we're dealing with. There's a relatively small set of locations where we have serological surveys that have been conducted. So we took these 21 locations where those surveys have been done, and for each of them, we estimated this force of infection due to natural infection after accounting for demography and vaccination coverage, like I mentioned earlier. And we see a wide range. There's a lot of uncertainty on some of these places. You basically get very low seropositivity. So that really is more of a reflection of our priors. But nonetheless, you see variation across multiple orders of magnitude in terms of what we're learning about these different locations. Now, like I mentioned, though, all of this rests on knowing whether these people are seropositive because they were naturally infected or because they've been vaccinated. And so, I presented that as blue and red earlier, but if you think about it as purple, and instead there's some probability that each of these people that are in each of these bars is either red or blue. And if we acknowledge that there's some uncertainty in that, that's one of the primary dimensions of uncertainty that we're looking to address in this work. The reason is that we, don't fully trust necessarily the vaccination coverage estimates. There's a lot of assumptions that can factor into them. In particular, for serological studies too, these 21 serological studies, a key thing that we have to rely on there is what they tell us about the subjects that they used in the study. Sometimes they say that these people were not vaccinated. Sometimes they don't specify. Sometimes they just sort of treat it as if it was a sort of random sample from the community in which you would reflect that, or you would expect that some proportion is vaccinated proportional to the coverage that we understand in that community. So what we make of these serological studies and the extent to which there's red and blue in here, we feel is something that we don't fully understand and something we want to explore uncertainty in. So to explore that, we came up with a set of 8 different scenarios about the set of assumptions we can make when interpreting these serological data. So one that I haven't mentioned yet, but that others have considered and we wanted to explore, is whether we exclude sites that where a serological survey was done following a recent outbreak. So you could assume that if these surveys are just done after an outbreak, that may not be a representative indication of a force of infection. It's a very selective sample. So we explored whether we include or exclude those. We explored whether we believe the vaccination status that's reported in a serological survey. We also considered the possibility that maybe it's just unknown in all cases, and so everyone has a probability of being vaccinated proportional to coverage across all the studies, regardless of what they said. We also explored excluding studies where where the vaccination status is unknown or unreported. And then we also considered the case where if some people say they're unknown, that they're just unvaccinated. Because again, I don't know about you, but it's difficult to keep up with these health records sometimes and vaccine cards and things like that. And so it'd be very understandable if people couldn't recall this accurately or if this information wasn't accurate in some sense. So we really explored all the possible extremes that we could think of about how to interpret these data. And just one thing to show you initially about this before we look at the implications more broadly. Here we're breaking down estimates of force of infection under these 8 different assumptions. So each of the different numbers along this x-axis is one of those 8 different assumptions I just told you about on the previous slide. And then these dots and lines connecting them are showing different force of infection estimates for different sites. So the more sort of vertical movement there is across the x-axis on these plots, the more sensitivity we're seeing in those assumptions. So we see in particular that 2 of these scenarios or sets of assumptions can pretty drastically alter what we infer about force of infection at some sites. The reason is that these assumptions have sort of the most impact on whether we assume that seropositivity is due to vaccination or natural infection, and they affect sites the most where the vaccination coverage is highest. So some places the coverage just isn't very high to begin with, so these assumptions don't really matter, but others it does. So we are seeing that there's quite a bit of variability that we get depending on those assumptions. So, and from here on out, I'm going to tell you more about how that uncertainty and how that variability sort of propagates downstream. Okay, so to explain though sort of where we're going with this, it's important to understand how the rest of the process works. So the next step in this process is to take our estimates of force of infection at these sites and then compare that to what we know about reported cases and deaths at those sites. So the way that this works is we're really trying to get an estimate of the degree of underreporting of cases and deaths that we can then apply to other locations where we don't have the serological information. So if the force of infection implies a certain relatively high number of infections, and if we observe this many cases and deaths, then that gives us information about what the multiplier is between cases and deaths and infections. So that's basically what we get out of this step in the process. And just to give you a sense of what that looks like, the x-axis in all of these is the same. Each panel is one of those 8 different scenarios that I showed you earlier. And you can see that some of these sets of assumptions, some of these scenarios give somewhat similar results, but others are very different. So notice here we have very low estimates of the proportion of infections that are observed, whereas here they're relatively higher. All of them are fairly low, but some of them are really low. So there's quite a bit of variation in what we assume about the degree of underreporting based on how we interpret those serological data from those studies. All right, so given that we've still got these 8 different scenarios, it could go any which way in terms of the degree of underreporting. The next step is for each of those 8 scenarios to then use those to basically take the cases and deaths that are reported in all of these administrative units across this region in Africa, including whether there are 0 cases or deaths reported, and then use that in combination with these estimates of underreporting from the previous slide to then extrapolate up to how many infections there might be in each of these places. I'll spare you any further mathematical details or plots of this, but that's just kind of the basic idea. And this map and the colors therein show you some indication the darker areas are more hotspots where it's suggesting that there's more infections, and we'll see more about this later. But because that simple extrapolation step, that's just really multiplying up essentially the number of cases and deaths that are reported. So we assume that that's going to be relatively noisy. And so that's why we take an additional step here to use regression models to try to smooth out that noise in a way that might be explainable based on environmental conditions. Again, yellow fever is a zoonotic disease. There are certain primate species that are involved in maintaining it. There are certain mosquitoes that are involved in vectoring it from those primates to humans. All of those things have different conditions where they thrive or don't. And so we take into account a variety of factors. We have variables on non-human primates and their distributions, temperature, precipitation, elevation, forest cover and forest degradation, other vegetation indices, whether an area is tropical, and then the extent of urbanization and things like that. So we try to use these suites of variables to try to smooth out those patterns in the projected infections in a manner that's hopefully reasonable. Now, getting into another set of assumptions, this is again a place where we sort of encountered a sort of a decision point, and we said, well, what type of regression model do we use? There's plenty of regression techniques that are out there. And so rather than just saying, oh, this is our favorite one and we're going to commit to it, we wanted to explore yet again the uncertainty or alternative possibilities of which regression models we might use. So again, we have a set of 8 different alternatives here that we explored. all the way from the simplest, where we have just a constant coefficient that represents our force of infection, to the next step, which would be that sort of intercept plus linear effects of those variables I described on the previous slide, linear and quadratic, various spatial techniques, and then a couple of more sophisticated machine learning techniques that would account for complex relationships among the variables and things like that. So just to briefly kind of give you a sense of that, all of these models, you can see there's some ability to explain some of the variation in our extrapolated or projected infections. So they all seem, potentially somewhat reasonable. I think based on this or other statistics, you can't just sort of, easily rule out some versus others. And so we wanted to proceed a little further and think more about, well, how do we take these different models and combine them or select among them? And so to do that, we decided to borrow a technique from another paper that applied some ensemble modeling techniques to mapping malaria and developed some nice methods for doing things like this. So if we start at the top with our 8 different regression models, what we did is that we withheld 10% of the data from fitting. So we just fit each of these models to 90% of the data. We did so in a way where we held out data at the country level. So it wasn't just random subsets, it was entire countries that were withheld from the models. So that made it a little bit more of a challenging test to then go and see how those models fitted to 90% of countries essentially did in explaining data from the other 10% of countries. We did that and we found that some models worked better than others. And then we basically ended up with an ensemble that consisted of the best performing combination of individual models in terms of predicting that out of context fit. And so that ensemble model is really just a weighted average of the predictions of those individual models. Okay, and so just as a visual to put on that, so if we have in total 64 different models at this point, so we're carrying around a pretty heavy load of models. We've got 8 different regression models, 8 different assumptions we can make about the serological data. So what I just showed you with that ensemble technique, we were able to kind of integrate across that uncertainty in regression models and end up with one sort of ensemble regression model per serological scenario, but we're still left with those 8 models. So unfortunately, we still have these 8 models and there's not a good way to combine them. The reason is that we can't rely on the same ensemble modeling techniques because they're ultimately informed by different data. And so there's not a sort of even playing field to compare them on. So this is a bit of an ongoing challenge for us. And this is actually something that the student I mentioned earlier, Quan Tran, is working on right now, is figuring out some creative ways to sort of integrate across this uncertainty, namely by trying to learn about the degree of reliability in reports of vaccination. So if we can understand that, let's say 70% of the time, we have a true positive and 30% we have a false positive in terms of somebody reporting their vaccination status, we can then apply that information to the vaccine coverage estimates or what was reported in a serological study to try to get more of a probabilistic treatment of what the real vaccination coverage might be, rather than sort of looking at these 8 different extreme scenarios. But nonetheless, for now we have these 8 alternative models. And one of the things we're interested in seeing is just how much of a difference does it make? I showed you at the very beginning of the talk different examples where there were different models that made very different predictions. So how different can things be in this particular problem? And so in general, we find about 100-fold difference in our estimate of the overall burden of cases and deaths across these different scenarios. So this is just sort of a few different ways to look at it. So projecting over time, looking at either deaths annually without vaccination or with vaccination, or deaths averted. Really, if you focus here, I think that's really the main takeaway. This is cumulative deaths from 1980 to 2030, so a relatively long time frame. And if we look across here again, we see that roughly 100-fold variation in these estimates of burden. So, this, you may think, well, this is just kind of uncertainty that we don't have and that we just kind of have to deal with and acknowledge. But I will tell you that this work is being done as part of the Vaccine Impact Modeling Consortium, which provides information to Gavi about investments in vaccination. And I will say that when we present information like this to them, they find it challenging to deal with, especially because they are making decisions about how to invest in vaccines across a large portfolio. They're not only interested in yellow fever, they're interested in other things too. And so if there's a 100-fold difference in the burden of yellow fever, that could have really big implications for their decision making about whether they invest in yellow fever if the burden's on the higher end, or if they think that yellow fever is a little bit less of a problem, and then they invest more in a vaccine for some other disease. So it is important, I think, to first of all acknowledge this uncertainty and not be blind to it, but again, to find new ways to deal with it and to try to refine it, whether through fancy modeling techniques or ideally through additional data collection or both. And again, I'll just plug that Juan is actively working on this problem, and I think he'll have some exciting stuff to report on this in the future. Okay, so moving on to the next part of the talk. Again, that was more of a spatial mapping context. Here we're thinking more about prediction and sort of future prediction and forecasting in time. So this work was led by Rachel Oitman, who's a former PhD student. She now works at an organization called Colorado Access, doing some really interesting work. And this was supported by initially an internship for Rachel at the UNICEF Office of Innovation there in New York City with you guys. And then also by an NSF Rapid grant that was supporting some work by us during the height of the Zika epidemic. Okay, so just to remind you, with Zika, this was a big deal back in around 2016, and it showed up in Latin America. We knew that Zika existed. It wasn't a completely new virus, but it was making its first introduction to the Western Hemisphere and really taking off. And there was a lot of uncertainty around that time about where things were headed, what the extent of the outbreak was going to be, what the time course would be, all these things. So, we've all lived through this very recently with COVID, and remember back to this time about 2 years ago where we had very similar questions on our mind with that pandemic. But again, the same sorts of questions were popping up with Zika just a few years before this. And so we're really interested in trying to figure out how do we respond to these types of basic questions and provide some kind of forecasts of where things might be headed in this context where there's so much uncertainty and where we don't have the luxury of having a long record of historical data to train models on. So that's really the challenge here, which again, I think there are a lot of parallels between what we're talking about here with Zika and what's happened during the COVID pandemic. And you all have some really expert leading forecasters on that front. So, but again, just to tell you sort of how we thought about this for Zika, and also to make a case for why we would want to at least attempt to make forecasts. And by the way, here you can be thinking about what I'm saying about forecasting. You can look at this while I'm talking about that, but this is just patterns of the Zika epidemic that we had the luxury of knowing after the epidemic. But this is what it looked like over the course of roughly a period of a year within Colombia. And each of these different rows is a different department or state within Colombia. So you see there's a lot of heterogeneity in the timing and the extent of the disease in different areas. So there's a lot of variability here that we'd like to predict. And some of the reasons we'd like to predict that variability or forecast it would be to prioritize the placement of vector control to mitigate the epidemic. This is, vector control, unlike, telling people to wear face masks or some of the things we've seen with COVID, where it's more of a bottom-up approach and you're just encouraging people to do stuff. You actually need to send out people and resources to apply these vector control measures to respond to the epidemic. So from a public health agency standpoint, it's really important to have some information to guide those decisions about how you allocate those resources. Also, again, similar to COVID, how much time do hospitals have to prepare for peak demand, and what is that peak demand going to be? Also, the demand for surveillance testing. Another issue which has come up with COVID too, it hasn't been quite as big of a challenge, but it is for a lot of emerging infectious diseases, is selection of sites for vaccine efficacy trials. So you want to make sure that you're going to set up your trial in a place where there is going to be a sufficient incidence of the disease to power your trial. And if the epidemic is going to be on its way out or it never gets big enough, then you're not going to be able to do that. So having some expectations to plan around is really important. And of course, people are just curious and want to know what's going to happen. Okay, so like I said, a particularly unique challenge in this setting for any emerging pathogen is that we don't have historical data to train on. Here's an example of flu data you know, there's a lot of variability in here and it's a challenge to predict, but nonetheless, you still, you know, pre-pandemic, you knew it was going to occur around the same time every year, and you kind of had a pretty good expectation and some past record to look at. That's just not possible for a situation like an epidemic of a newly emerging pathogen. Also, the quality of the surveillance data, it is always poor for just about any infectious disease, but it's especially poor for an emerging one where people aren't even, the disease isn't even on anyone's radar initially, and then that's sort of quickly changing over the course of the epidemic, and the testing resources are not widely available. So a lot of challenges here. And then also another challenge in this context is that even though Zika was not unknown to science, it was certainly understudied. And there were a lot of things that we did not know about its epidemiology and just the pathogen's biology in a very basic sense. So we were sort of learning as we went. So there's all sorts of dimensions of uncertainty, assumption uncertainty that we might want to explore in this. So our approach for dealing with this problem was to use a discrete time stochastic model, which is shown here. So this is sort of a variation on the TSIR model or the renewal equation model. It's sort of in that genre of models. We accounted for spatiotemporal variation in the reproduction number. Again, this is a mosquito-borne disease, so we know that it's going to be sensitive to temperature and other weather type variables and things like that. We also accounted for immunity by making it an SIR-like model, so that essentially we would eventually we would expect there to be a downturn in the epidemic, probably due to the buildup of immunity. So we have that feature in here. We also had connectivity between these different states or departments within Columbia, because of course, initially they weren't showing up in all of them at the same time. So it was important to be able to try to account for those initial patterns of spread. And then the way that we went about parameterizing the model was with a data assimilation approach, where we start out with some prior assumptions, and then we have the model learn as it goes based on how it's performing in forecasting. Okay, so just to walk you through a few of the assumptions and kind of what they look like in the model. So consistent with the theme of my seminar today, we explored some different dimensions of uncertainty in different assumptions. The first one was human mobility. So we had a few different versions of the model that we looked at there. So one was to use cell phone data, which we had available at the department level. This wasn't sort of a real-time cell phone data, unfortunately, but it was from a fairly recent time period. So it gave us a pretty good idea of what people's movement patterns are between different regions of the country. We also considered a simpler gravity model and a radiation model, which we don't need to inform with any particular data, but you know, provide two different pictures of what mobility patterns could look like. And then we also considered a model in which there was no movement among departments. So we just started forecasting locally within that department once cases showed up there. So I think this covers a pretty wide range of possibilities that are commonly used in different forecasting and spatial modeling exercises. Another dimension of uncertainty we explored was how we actually set up the spatiotemporal variation in the reproduction number. So we looked at two different possibilities. The one on the left built on some work that I did very early on in the Zika epidemic, where we came up with some ways to use temperature and mosquito occurrence maps and a socioeconomic indicator that was spatially resolved to get some estimates of the reproduction number. And so this is we refer to this as a static because it doesn't account for seasonal variation. It's just sort of a time averaged indicator. But you can see it does predict quite a bit of spatial variation in the reproduction number across the country. We also considered what we refer to as a dynamic formulation of spatiotemporal variation in the reproduction number. So this built on some work done by Aaron Mordecai and others, where they took curves that relate temperature to the reproduction number. And so that allowed us to have something similar to this in terms of capturing spatial variation in temperature, but also capturing the temporal component. So we would have different reproduction numbers in different months and things like that. But there are some slight differences between these two models in other respects in terms of what these temperature relationships are and what other variables account for it. So in any case, these are two very reasonable approaches that you could take. Either one would be justified. And both were actually information that was available at the beginning of the epidemic that could have realistically been used for such an exercise. And then finally, the last aspect of uncertainty that we looked at were the number of introductions. So whether there was just a single introduction that seeded the virus into the country or 2 introductions, we also looked at more. It didn't really seem like that was likely, but there was some debate based on phylogenetic analyses and other patterns about whether there were 1 or 2, which you could imagine could be important if the virus, or if cases started being reported on 2 different ends of the country initially, and then they kind of spread towards the center or something like that. So there were some, if you squint and look at the data, you could sort of think about it either way. So we wanted to explore both of those possibilities and see if it made any difference in how our models performed. So in total, this resulted in 16 models that reflected all these different combinations of uncertainty. Certainly, there's more uncertainty in our assumptions that we could have explored, but we felt that this captured a pretty decent range to look at. Okay, so over the next few slides, I'm going to show you a sequence of slides that look like this, where I'm trying to give you a sense of what the forecasts look like, how they changed over the course of the epidemic, and so on. And just to orient you a little bit, on the left, we're going to see individual model medians. So we're going to have a total of 16 lines on each of these plots that reflect a median forecast of each of our 16 models. You can't see many lines here yet, but you will see more as we go. And then over here, you're going to see what an evenly weighted ensemble would project. And then each of these panels is a different department within the country. So we're interested in projecting those separately. And the other thing to keep an eye on is what it says up here in terms of how many weeks of data have been assimilated into the model, with this pink bar in each plot indicating sort of the time of the forecast. So this is really what we would have expected based on our prior assumptions that we put into the model that weren't informed by these data, which, as you can see with these individual models. Oh, and by the way, the purple, those are reported cases that we eventually saw, but of course weren't used to inform the data. So early on at the very beginning, you see that most of these models are predicting hardly anything to happen. They're just not expecting a big epidemic, which actually reflects a pretty reasonable prior, because before the start of this epidemic, people wouldn't have generally thought that Zika was going to sweep across the Americas. They just didn't think that was a much of a possibility. And likewise, the ensemble model reflects that as well. So to make things a little more interesting, when we move ahead 12 weeks and account for the first 12 weeks of data in the epidemic, you start to see that the models are changing their tune quite a bit. Here we see all those different lines that I referred to, where these different models are making forecasts of somewhat different trajectories. In some places, things look pretty similar, but in other departments, things are more variable, which probably reflects a lot of the uncertainty about the spatial spread, because at this early point in the epidemic, it had not showed up everywhere. So it wasn't just a matter of forecasting the trajectory, but also forecasting the start of the trajectory in different departments at that point. So you see a lot of variability there. And likewise, the ensemble, you see that variability too. But if you notice this kind of faint green band, you see that even at that point, in a lot of places, that band is encompassing the trajectory of what actually unfolded eventually. So even though there's a lot of uncertainty here, it did at least encompass the eventual possibility in many places at a fairly early stage. But of course, that performance is better some places than others. When we give it another 12 weeks of data, by this point, we're almost at the peak of the epidemic. And you see that a lot of models are actually doing a pretty good job of recognizing the fact that they are at the peak and that things are going to go down from there. Although there is quite a bit of variation in some of these models in terms of what that trajectory looks like on the downside of the epidemic. In particular, there are some things like this example up here in Antioquia is a good example here, where you see like sort of two different sets of lines. And those actually reflect the different assumptions about spatiotemporal variation in R. So the one that's just sort of a straight downward line, which actually seems most consistent in this case with the data, that's where we just have a static R, where it's not changing over time. Whereas this one that's sort of bumping back up later on, That's because there's this temporal component of variation in R, and it's assuming that the weather is going to pick up and increase transmission, but that didn't really turn out to be the case there. So anyway, you see some discrepancies that are quite explainable based on these model differences, which is something that we were interested in learning about. And again, the ensemble is just a simple, evenly weighted ensemble is doing a pretty decent job of capturing the trend in a lot of these places. And then finally, give it another 12 weeks and it just sort of constrains those uncertainties, and the models tend to agree a little bit more, but also reflect some differences here. You continue to see in some of these departments the consequences of that temporal variation in R, that it's assuming it just knows that things are going to pick back up when in fact they're really not, because probably the trajectory at this point was largely determined by those immunity feedbacks. Okay, so to do some more, to try to do some more comprehensive comparisons, there's a lot of dimensions here, the different models, different departments, different times, but I'll try to summarize some of the things that we found. So there were a couple of different things that we found early in the epidemic versus late in the epidemic in terms of which models perform best. So on each of these plots, we have the different models, those 16 different models arranged here with basically the best performing model at the top and the worst performing at the bottom. Along the x-axis, we have the different weeks in the course of the epidemic. And then each of the three panels is a different metric that we're interested in forecasting. So the timing of the peak week, the incidence at the peak week, and then the week by which 10 cases occurred. So this would be sort of a prediction of when the epidemic sort of starts in some sense. And if you see a green color here, that indicates that an individual model performed better than the ensemble. And if you see a reddish purplish color, that indicates that the individual model performed worse than an evenly weighted ensemble. So there's a few patterns here that I'll draw your attention to. Things differ across the different metrics a little bit, but I think there are some consistencies. So in general, we see that there are a few models at the top that outperform that evenly weighted ensemble early in the epidemic. And those tend to be the models that have this spatial information, in particular, the mobile phone data. Those tend to perform pretty well in multiple regards at that early phase. And that makes a lot of sense because they are the ones that are really capturing that sort of invasion process and the spatial pattern of spread. So that information turns out to be really useful at the beginning. However, as we get into later points in the epidemic, once the virus is established in each department, it appears that that influence of those introduction patterns matters less because things have sort of taken off and on their own in each of those departments. So that's one thing that we found. And then also just in general, as things went, we found that the ensemble really performed better once things hit the peak. Again, that sort of drowns out a lot of that sort of those poor assumptions, especially about the temporal variation in the reproduction number. So another way that we looked at this, there's different ways to combine models and ensembles and things like that. So this was a bit of a retrospective analysis here where we looked at what sort of an optimally weighted ensemble would have been at different phases in the epidemic. So this is less about sort of, how we go about doing forecasting, but more about making some inference about which factors played an important role. And so again, kind of like what I talked about on the last slide, we see those more refined models spatially, they turn out to be more heavily weighted early in the pandemic or the epidemic. But then later on, once it gets more into those just sort of internal dynamics within each of the departments, and especially around the peak, you see that models without any movement do better. They're sort of ignoring the noise of those introductions and really just sort of capturing those sort of internal dynamics. With the number of introductions, we see that that's a bit of a mess, and we don't have a very great understanding at any point about what that looks like. And then the other thing is, like I mentioned, that at some points in the epidemic, we see performance of models with the static versus the dynamic R formulation differing somewhat. The dynamic model does play an important role at some points around the peak, but then less so later on and earlier on, where you really just need to sort of know the magnitude of R naught, especially at the beginning, rather than all these temporal fluctuations. Another thing that we did was we looked at that evenly weighted ensemble on the right, which is just the simplest way to aggregate these forecasts, versus this weighted ensemble where it was a more optimally tuned way to do things. And we found that really, on average, the evenly weighted ensemble was a better bet. So even if we did have the ability to kind of do a sophisticated weighting scheme, at least the weighting scheme that we used didn't really provide a consistent advantage, and this evenly weighted ensemble turned out to be pretty good. However, I will say that I know that especially during the COVID pandemic, that there's been a lot of progress in coming up with ways to weight different model forecasts in a real-time manner and stuff. So there are probably better ways to do this now, but at least based on the tools that were available to us then, it looks like the evenly weighted ensemble is probably a better way to go. Okay, so just to wrap up on this part of the talk. So there are a lot of different assumption uncertainties about emerging pathogens that make forecasting them difficult, but ensemble modeling, at least based on our experience here with Zika, does appear that it can provide an effective way to hedge against those different uncertainties and to come up with a single prediction that can be sort of focused on. And we found that data assimilation was quite effective at getting our models to sort of change their tune as they went. Like I said, they wouldn't have expected an epidemic to occur at all at the beginning, but they kind of quickly got up to speed and they were able to predict the peak pretty well, and then what happened on the downside of the epidemic as well. Now, I think some ongoing challenges from this. One is that, you know, I'm advocating here for an evenly weighted ensemble, but at the same time, if you throw a bunch of models that are too much like junk and that aren't good enough, then of course that's going to make the ensemble suffer. So I think that it's really important to try to use exercises like this to learn as much as we can retrospectively about which models perform best in these contexts, so that we can then make more principled decisions about which models we might include or consider in the future. Because ultimately things are going to, any sort of average is going to be a reflection of the individual models that go into it. So if we can come up with better models to get to sort of increase the average overall, that's ultimately what we'd like to do. So I think that, again, this is an ongoing challenge, and I know the focus of a lot of work with similar ideas in the COVID pandemic. So just to wrap things up across both of these parts of the talk now, I'll just highlight a few sort of overall conclusions. There are a lot of forms of assumption uncertainty. Again, they could have to do with the biology, such as how spatial temporal variation and reproduction numbers come about, different mathematical choices, or different assumptions we make about the data, such as the how we interpret that serology data in the first part of the talk. Addressing assumption uncertainty really often, I would say, just boils down to checking our models. So if we just pose models with different alternative assumptions and compare them, that's a really important starting point. And it's important to simulate under those alternative assumptions and then validate those inferences. So we really want to be open to multiple different models and sort of checking them throughout any of these processes or exercises like this. I think there's definitely an important role for ensemble modeling to try to reconcile these differences and to try to come up with an aggregated estimate. Like I said earlier, if we present a decision maker like Gavi with too many different models or too wide of a range, it can be difficult for them to know which part to focus on. So I think there are some important ways that ensemble modeling can help address that. And there are a lot of, there's a lot of progress in different techniques for doing that I think is worth paying attention to. And then the last message I'll say is that while we've tried to pull out whatever modeling tricks we can to get around these problems and to mitigate this assumption uncertainty, there's no substitute in many cases for better data. There are a lot of data gaps out there, especially in the yellow fever part of the talk that I spoke about earlier, and just inherently when you're dealing with an emerging infectious disease like Zika. So it really is important to, I think, take the models as far as we can, show how far we can get with them, and try to use them to justify the collection of different data, or at least show what the value of that would be. So with that, I just again want to thank my lab and the many people who contributed to different aspects of this work, as well as the funders for these and other projects in the lab. So thanks. I'd be happy to take any questions if there's time.
Thank you so much, Alex, for the wonderful talk. It's very interesting, very important work. So we have a few minutes for questions from our audience. So there's one question in the chat, so I can bring up to the audience and also Alex. So could the speaker please help me to understand the individual versus evenly just weighted ensemble, the Zika work? I might miss something silly. So
Yeah, sorry, maybe I'll share my screen again.
Yeah.
Yeah, I probably don't have sort of like an ideal slide for that, but I'll try to maybe work on this. So here, if we have each of these black lines is the median for a different single model. So we have 16 different lines here. In addition to that, there's some band of uncertainty that's not shown here. So the evenly weighted ensemble is really just a simple matter of sort of evenly combining those 16 different distributions to form one composite or ensemble distribution. The alternative way to get an ensemble that's more sort of optimally weighted, this was based on some approaches that Nick Reich and others had used in a paper a few years ago. It's basically based on performance with respect to different forecasting metrics at certain points in the epidemic. So it's really sort of trying to more heavily weight models that perform better, just to put it simply. So,
Jeff?
Hi, Alex. Very nice talk. Thanks for joining us for this. I am a little curious about your conclusion that the evenly or straight average ensemble does better than the weighted ensemble. That's really typically not the case. Do you think that's just a function of the fact that you're really only looking at a single outbreak over this time? And also, I'd point out that the advantage of a weighted ensemble is that it does meet out the models that are not performing well. It automatically provides you, well, not automatically, it provides you an objective framework for discounting models that have poor performance. So you do not have to concern yourself as much about sort of any sort of subjective screening of models beforehand, and allows you to include things such as a uniform model or very simple statistical constructs that may actually have worth at some level in the ensemble.
Yeah, so I think I would not go too far with that conclusion. I think that's what we found in this particular case. But I think we probably could have pushed it farther. And like I said, I think there's better techniques than what we use probably for doing this. Another reason we may have found that, though, is the variation in our models is probably not as pronounced as it is in some cases. These were all quite related models. They were really from kind of the same family. If we had used a wider range of models where the variation in performance was even greater, I think maybe some of the principles that you mentioned would have been more important. So yeah, I wouldn't take it too far, but at least in our case, I think that's what we found. And we also found, I think over the time course of this epidemic, it's just an up and down sort of thing. There wasn't really a long time frame to assess this. And again, what we found, though, and this is maybe the key thing about why we go to that conclusion, is that when we look at what the optimal weights are for those models at different points in the epidemic, it was changing. So we could have predicted that, or we could have gone with the initial weights, but a few weeks later, we should have gone with something different. And again, I think maybe it's just the speed of the epidemic and the nature of that particular one. I'd love to have better ways of weighting these ensembles, and I probably need to read up on some things, but that was our experience anyway.
Thank you.
Any other questions from our audience? No? Okay. So Alex, I have a question. So you mentioned that when you do the Zika work, you look at the initial transmission model. You found that models incorporating mobilities across different locations will give you a better prediction than the ensemble model. But after the peak, the ensemble model performs better than the individual models. So do you think, if we have a different objectives for the prediction, like I want to predict the spatial movement of the disease, maybe we can use a network model you proposed, incorporating the mobility, but after the disease takes hold in the community, maybe mobility not that important, we can just use the local model to do the prediction. Is that something you think is reasonable?
I think that's consistent with our results. And I think that's, in general, that sort of thought process about which metrics do we care about at different points and what models are going to be most appropriate. I think that's really largely what we're advocating for and trying to do, is to try to better understand what are the principles or the guidelines that we use to make those types of decisions to use a better set of models or a better way of combining them or evaluating them at different points. So yeah, in retrospect, it's not that surprising, and we probably just should have known that. But I think our results certainly demonstrate that that was the case in this example.
Okay, thank you.
Yeah, I see there's a question. Yeah, about whether the code for our work is open source. I believe all of it is. If you go to my lab's website, we have a page with publications, and next to the link for all the papers, there's generally a link for the code. If you can't find that or anything, feel free to email me. It should be listed in the papers too, but yeah, thanks for that question.
All right.
Yeah, I think we are approaching the end of the seminar. So we thank Dr. Alex Perkins again, and have a good day.
Thank you all. Bye.
Take care.